Hello, this is Greg Ristabin from Olympus. I'd like to welcome everyone today to this first webinar in the Logos and Olympus webinar series titled Introduction to Sample Clearing. Today's topic is on imaging optically cleared samples. In this first part of our series, we will discuss optical clearing of biological samples and its benefits. We will then discuss how these samples can be imaged using various microscopy techniques and the advantages and disadvantages of each method to help you determine which system is best suited for your needs. Our presenters today are Tim Stepp and Gulpreet Kaur. Dr. Stepp is an application specialist with Logos Biosystems. He has a PhD from UNCCH in marine sciences and 15 years of industry experience. Dr. Kaur is a national representative for the Olympus Scientific Solutions team, specializing in the Alpha 3 Lightsheet system, and helps scientists apply cutting-edge imaging technologies in their research projects. This informational webinar has been scheduled for approximately 1 hour. If you have questions, please type them into the Q&A panel during the presentation. If we're not able to get to your questions during the webinar, they will be addressed personally either by email or phone after the event. Now, without further ado, I'd like to turn the presentation over to Dr. Gulpreet Kaur. Dr. Kaur, take it away.
Hello, everyone, and thank you for attending this seminar on sample clearing. I'm Gulpreet Kaur. I work with Olympus as an application specialist focusing on high-end imaging systems such as spinning disk and Lightsheet microscopy. Now, as microscopists, we do tend to focus a lot on new and exciting imaging technology. But with this seminar, we would like to shift the focus from imaging cool samples to preparing cool samples by clearing them. With this in mind, we have with us Dr. Tim Stepp, application specialist with Logos Biosystems, focusing mainly on sample clearing technology. This is the first seminar in our series on sample clearing, so we would like to start by introducing sample clearing. This will make up for the first part of our talk today. Then we'll have Tim discuss some of the newer advances in tissue clearing. Finally, I will talk about advances in imaging technologies that allow one to image large cleared samples effectively. And then we will discuss some considerations while imaging these. Now, a key concept in biology is the correlation between structure and function of life forms at various levels of organization. This means that one can understand the function of cells, tissues, organs, and organisms by studying their structure in detail. For example, this video shows vasculature in a whole mouse brain. From such data, you can study several factors, such as regions where the density of vasculature is higher compared to other regions of the brain, and then you can further deduce the reason for the same. Now, as all life forms grow and develop in three-dimensional space, it is critical to capture imaging data in X, Y, and Z planes with high resolution. However, as thickness of the sample increases, it becomes increasingly harder to image through non-cleared samples. It might seem trivial, but have you wondered why we can't simply image through some samples? Why can't you simply take a leaf from a fluorescent plant and image through it as shown in this image? This is because incident light enters non-cleared tissues, and it is immediately either scattered or absorbed by different cellular components. These include membranes, ribosomes, nuclei, mitochondria, myelin, or even cytoskeletal components. Sample clearing removes the major light scattering and absorbing molecules in these cellular components and permits light to pass through the sample efficiently. Thus, the entire depth of even a thick sample can now be visualized easily. Traditionally, voluminous samples were imaged by serially cutting the sample and then trying to reconstruct this whole image. However, this method is time-consuming and generally unreliable. Newer advances in tissue clearing and imaging technologies have permitted us to image large samples with high resolution and create data sets such as this movie of a whole fly head. There are many protocols for clearing different types of samples. New and complete systems are now available to help researchers produce produce reproducible clear tissue samples for imaging. Now, Tim will discuss the X Clarity system as well as considerations for tissue clearing.
Thank you, Gulpreet. Since 2015, we have produced the X Clarity system. And before that, I do want to thank Olympus for hosting this webinar. And we especially want to thank all of you for your time and attention. And since we've released the X Clarity system, we've had the privilege to speak and collaborate with researchers around the globe as they successfully established reliable and reproducible tissue clearing workflows. Many, we are proud to say, have chosen to integrate the X Clarity system in their process. For others, the X Clarity was not necessarily the right tool for the purpose, but all have been gracious in providing their time and feedback. As a result, we would like to share some of what we've learned along the way in hopes of helping you establish your own successful tissue clearing workflow. However, before we get too far down the road, for the purposes of this discussion, we do want to establish a couple of simple functional definitions for tissue clearing, both in terms of what it is and, more importantly, what it does, or the end result, at least with regards to clarity-based clearing methods, which, if the name didn't give it away, then the X Clarity system falls into that category. Tissue clearing is a two-step process. The first is removing, and the second is replacing. The first is removing the major light scattering discontinuity source, which are lipids, and the second is replacing the intra and extracellular water with a refractive index matching solution. The end result, or what tissue clearing does, is that it creates an intact, optically transparent tissue sample that can be imaged at depths over several millimeters to up to potentially to a centimeter in depth. However, fundamental to establishing a tissue clearing workflow is understanding that while it is an important component, it is just one part of a larger analytical pipeline designed to acquire, analyze, and interpret in-depth three-dimensional images. In fact, from our experience, the other parts of this analytical pipeline should be planned and accounted for, if not prior, at least in conjunction with establishing a tissue clearing strategy. This is because these factors can significantly influence your overall tissue clearing strategy by determining the volume or size of the tissue that should effectively be cleared, and also by influencing the actual tissue clearing method that you choose. And as you can see, and why we've teamed up with Olympus, is that the imaging system plays a major component in this. And after my portion, Gulpreet will also go further into the considerations for imaging and using the different systems. Therefore, our goal in this portion of the discussion is to help you understand how these other components relate to tissue clearing and how to optimize your overall tissue clearing strategy based on that information. We hope to do this by posing some questions and making some recommendations along the way that we think will help you in your journey to establishing a reproducible tissue clearing workflow. Certainly, we'll focus on optimizing a pipeline using the X Clarity system. However, most of what we will be addressing is applicable no matter the tissue clearing method you decide to employ. When we have our initial conversations with those who express interest in the X Clarity system specifically, or just about tissue clearing in general, our first goal is generally to try and understand your overall research goals and how you think tissue clearing will help you achieve those goals. So our first questions revolve around why. Why do you want to clear tissue? How do you think it will benefit your research? What insight do you think tissue clearing will help you obtain? Rarely, but on occasion, we do hear responses like, we want to take cool images, or our department bought a Lightsheet microscope and we want to use it. Mostly, though, we hear well-thought-out responses like, we want to know how BEN1 deposition relates to amyloid pathology in Alzheimer's models. We are trying to map and develop alternative neuronal pathways to aid recovery from a spinal cord injury. We want to map alterations to bone marrow vasculature and Crohn's disease model. Or we want to better understand the role of alternatively activated macrophages within glioblastomas. As Gulpreet mentioned earlier, we live in a three-dimensional world. Therefore, to better understand the structure and function of our model systems and how disease or environmental factors impact these systems, we need to be able to better visualize and analyze these models, not only with a three-dimensional context, but across different size scales. Simply put, we need to be able to see both the forest and the trees at the same time. Tissue clearing helps us do just that, see both the forest and the trees at the same time. So for our first set of recommendations, include to be sure that you have clearly defined goals and questions, understand the role of tissue clearing in the larger 3D imaging analytical pipeline and how tissue clearing can help you achieve those goals, and just as importantly, how tissue clearing cannot. Similar to RNA or DNA extractions, tissue clearing is a sample preparation method. A sophisticated and involved sample process to be sure, but a sample prep method nonetheless. Just as the quality of RNA or DNA will determine the success or failure of downstream analysis like sequencing, the quality of a cleared tissue will determine the quality of your downstream imaging. However, unless you have a microscope system that can perform in-depth imaging, it won't matter how well your tissue is cleared. So our next areas of conversations generally concern what your available imaging systems are, because if you don't have an imaging system such as a Confocal, a Multiphoton, or a Lightsheet, or an objective set that has a sufficiently high numerical aperture and deep working depth, it really won't matter. Does it mean that you can't obtain these 3D images? It does mean that you will need to make arrangements to access somebody else's or even perhaps purchase a system that is capable of this. Each of these three systems has its pluses and minuses with regards to imaging depth, resolution, and scanning time. Gulpreet will provide more detail about the different systems a little later. From our perspective, with regards to imaging systems, one of the best pieces of advice we think that we could provide, and we hope that you would follow, is that talk to your imaging specialist. Your imaging specialist for your institution or department will be invaluable to your efforts. Share with them what you want to do, find out what is available for you to use, and seek their advice for their best approach to take. The second is to understand the refractive index, depth limits, resolution, and data requirements of your system and objectives, and plan accordingly. So, for example, in a hypothetical situation, let's suppose that your goal is to map the distribution of Kupffer cells within an entire mouse liver in the context of some disease or a toxic exposure. The imaging system that you may have access to is a confocal microscope, and the objective is a 10x air objective with a 4-millimeter working distance. Given the size of a whole mouse liver, imaging an intact liver with a confocal would not be practical due to the scan time and depth limitations. This doesn't mean that you cannot obtain significant and informative images or even build an entire 3D construct of the liver. It does mean that you will need to plan your approach, either use smaller sections or reduce the total volume of tissue that you will be imaging. While I don't want to give away too much, one of our recommendations with sample volumes down the road. The third is to make sure that your lab or institution has the necessary data handling and analysis capabilities to handle these data sets. These are extremely large data sets, from high triple-digit megabyte sizes to low double-digit terabyte sizes. And so the data handling, transmission, and analysis requirements are not trivial, but for our purposes, they don't really touch on what sort of tissue clearing strategy we have. So just be sure that this is one of the things that you do want to find out about, is if you can actually handle and analyze the data. After we discuss the imaging system, then we move on to more detailed discussion around the tissue type to be cleared, features that you want to capture, and the volume that you will need to adequately capture those features. Tissue clearing came to prominence for its ability to clear neuronal tissue. However, it can be used for a breadth of other tissue types. One of the most frequently asked questions about tissue clearing takes the form of, we are not working with mouse brain, but we are working with, say, kidney, liver, spleen, etc. Can we still clear tissue? With the X-Clarity system, at least, the answer is yes. Yes, you can. Further, clearing is not limited to mouse. It can be formed with other organisms, including rats and even possibly fixed human samples. Certainly, some tissue types are more recalcitrant to being made optically transparent than others. The older, more pigmented, fibrotic, or fatty a tissue is, the more difficult it will be to clear. You can still use tissue clearing with these samples to image depths significantly greater than non-cleared tissue, but you will need to determine the optimal sizes that can be cleared. So if we go back to our example of imaging a whole mouse liver, suppose that rather than a confocal microscope, you have access to a light sheet, which potentially could scan and image a whole mouse liver in a reasonable amount of time. And you're studying a fatty liver disease model, and the mouse is relatively old, say 12 to 15 months. While the light sheet may provide the ability to image the entire liver, the amount of fatty tissue and fibrotic tissue that is accumulated with the aging may limit the size of tissue that can practically be cleared without substantially altering the tissue's structure. While the unofficial clearing reference standard may be a whole adult mouse brain, tissue clearing is not just for mouse brains. Just know that the tissue type, composition, and age will impact clearing efficiency. For example, with an X-Clarity system, an 8-week-old adult mouse brain could be cleared in as little as 7 hours. Conversely, 12 to 15-month-year-old mouse brain can take twice as long and may not be completely cleared. And then with this set of recommendations, and we've bolded it out for a reason, is we cannot emphasize this enough, is clear only the volume of tissue that is necessary to capture the features that you want across the size scales that you need. Yeah, one of the features of tissue clearing is it can allow you to image a whole organ. However, if you don't need to clear a whole organ, then we encourage you to save your time and resources and clear only the volume that is needed. Our next questions will revolve around what sort of labeling scheme you will be using for the specific analyte you want to label. This includes antibodies, proteins, fused with fluorescent molecules like GFP or RFP, and fluorescent dyes like dextran. Here too, the choice of analyte and labeling method can influence the scale of tissue to be cleared and your clearing method. By far, the main analyte target are proteins with other targeted analytes, including lipids, nucleic acids, and macromolecular structures using specific fluorescent dyes. You will want to check the compatibility of both the analyte and labeling agent with your method of choice. As another example, if you're using a dye that binds to lipids, you will not want to use a clearing method like a clarity-based method that removes lipids. Nucleic acids also may be targeted through counterstains like DAPI or sequence-specific probes for RNA and DNA. Counterstaining is compatible with most clearing methods, while to date, in situ hybridization has only been used in conjunction with aqueous-based methods. The two main means of visualizing proteins is either through standard immunohistochemistry techniques like using antibodies or by virally or endogenously labeled fluorescent markers like GFP to a protein. As we'll show here in a little bit for the X-Clarity system, your labeling scheme, at least with regards to proteins, is the major component for setting the parameters when you actually cleared your tissue, but we'll get to more of that here in a little bit. With regards to antibody labeling, just about all clearing techniques are compatible with antibody labeling. With some techniques, such as solvent-based clearing methods, antibody labeling is performed prior to removing the lipids. while others, such as clarity-based methods, antibody labeling is performed after the clearing. For example, for samples that will be antibody labeled, we recommend that you overall limit the size of your tissue sections. Antibody labeling can take a considerable amount of time and significantly stretch your consumable budget. For example, with a whole mouse brain, labeling can take up to 4 weeks per antibody. Other organs would take even longer, during which time you may experience some degradation of tissues, and even with a high concentration of antibody, the distribution of labeling through the entire sample may not be even. There are penetrant kits that can improve the distribution of antibody labeling within larger samples. Logos even offers a deep label antibody kit that can improve labeling times, antibody distribution, and specificity. Nevertheless, especially when you're first starting out, we recommend that at least for brain samples, you keep the size to no larger than a mouse hemisphere. For denser tissues like heart or liver, we recommend you restrict the size to no more than about 2 or 3 millimeters. If your tissues will contain an endogenous or virally labeled protein, then there is no real limitation on sample size that can be cleared, other than what your imaging system dictates. As with antibody labeling, the vast majority of clearing techniques are compatible with endogenous labels, although aqueous-based methods generally will yield better images for endogenous for samples. So for labeling, we have a couple of different recommendations. One is be sure that your target and labeling agent are compatible with your method, okay? So it's not going to help that if you are, say, again, want to target your lipids within a membrane, say, and use a dye that specifically targets those, and you use a clarity-based method, that's unfortunately going to not show anything because the lipids have been removed. The second is start small. You'll hear us mention start small with regards to clearing overall. Before trying to label tissue sections, we recommend that you start with 1 to 2 millimeter thick sections for antibody labeling to at least get an understanding of best concentrations and the overall time it may take to label. Starting small will also allow you to more easily optimize and minimize potential waste. Overall, in general, we'd recommend that if you want to visualize a whole organ, if possible, that be virally or endogenously labeled rather than trying to rely on antibody labeling for something as large or similar in size to a whole mouse brain. So now you're ready to decide what is the best clearing method for your purposes. The good news is that there are a lot of methods to choose from. The bad news is just the same, as there are a lot of methods to choose from. So you've likely seen a graphic similar to this that depicts all the various clearing methods based on their underlying chemistry. Given that tissue clearing will be increasingly employed in the future, this chart will likely grow as innovation continues. There's a lot of literature out there that compares and contrasts the different methods, so we'll not get so much into that. We'll simply say, for now, that all tissue clearing methods follow a basic workflow with some difference on when, if it is needed, antibody labeling occurs. And then for now, we will also simply, and let it suffice to say, that all methods have their pluses and minuses. Certainly we have our opinion, and it's not difficult to figure out what that may be. Ultimately, though, the question for you is not what is the best clearing method overall, but what clearing method will be the best for your purposes. So at this point, for those not familiar with, we will introduce you to the clarity method and X-Clarity system, and then come back to our recommendations for determining whether the X-Clarity system is something that for you to consider. The clarity method takes advantage of the fact that a hydrogel, in this case acrylamide, can be infused through a biological tissue and then polymerized. The polymerized hydrogel acts to maintain a tissue's macrostructure. The lipids are removed either passively or actively using a clearing buffer made of SDS. Passive clearing simply refers to soaking your sample in a solution or series of solutions, whether you're doing this by the clarity method or any other type of method. Active clearing refers to clearing by applying an outside force to speed up the process. In the case of active clearing with regards to the clarity method, an electrical current is applied to the sample. So, for example, to tell you the difference between active and passive clarity is in the passive clarity method, a whole adult mouse brain may be cleared in weeks to months. While the X-Clarity system, which is primarily an active clearing system, a whole mouse brain can be cleared in as little as 7 hours. Which brings us to the X-Clarity system itself. X-Clarity system is an automated turnkey system designed to facilitate active clearing when using the Clarity method. Clarity method, which was first published by the Dice-Roth Lab at Stanford in 2013, in 2015, Logos licensed the technology from Stanford, and we released the first generation of the X-Clarity system. In 2018, we released the current version that's on the market today. For those of you familiar with the passive clarity process, you know that while it may take some time and that it is an easy and reliable method to clear tissues, the X-Clarity simply makes an easy process and drastically reduces the time. The X-Clarity tissue system is comprised of the X-Clarity tissue clearing system, these two components right here. This is the clearing chamber where your actual sample goes. This is the control tower right here. In the back, it may not show up on the white background so much, but is the buffer reservoir where you put the SDS buffer. And you have tubes that are connected to the clearing chamber and also to the built-in peristaltic pump within here. The second part of the automated system is the polymerization chamber itself. This is simply a one-button touch, 3-hour polymerization, so you don't have to set up a desiccator or flush with nitrogen and anything else. The other parts of the X-Clarity system include the consumables. The hydrogel, which is a 4% acrylamide formulation, the clearing buffer, the antibody labeling kit, and also the refractive index mounting solution. We also have several different types of sample holder that range from a single sample holder all the way up to 192 small sample holder for, let's say, biopsies or something like that. But by far, the most popular and most flexible sample holder that we likely have is the 6-mouse brain holder. Call it the 6-mouse brain holder, but it'll hold samples that are equivalent in size. So it'll hold generally most other, any sample the same as a mouse brain. Similar to the chart that I showed earlier about the overall tissue clearing workflow, the X-Clarity workflow follows from animal prep, perfusion if you're working with mouse or rats, and sample extraction. And so, although perfusion is the best method because what that does is it washes out the heme and blood, which can interfere with the downstream imaging. But it doesn't not preclude you from actually using archive samples that may have been fresh frozen or fixed in formalin ahead of time. The second is a sample fixation step, or what we call the post-fix step. And this is once the tissue sample has been extracted from the animal, is you simply incubate this at 4 degrees in a 4% PFA solution for 24 hours. At this point, samples may be stored for up to 3 months. After sample fixation, and certainly if you're working already with prefixed samples, you don't need to do this secondary post-fix. The hydrogel infusion, where you incubate your samples in the hydrogel to allow the acrylamide to infuse or diffuse throughout the sample. And that also goes for 24 degrees, or 24 hours at 4 degrees. And then comes the hydrogel polymerization. And with our polymerization system, as I said, this is simply a matter of placing your samples in 50 ml tubes or 24 well plates, closing the lid on the system and pressing run. And 3 hours later, the samples are polymerized. The next is the actual tissue clearing. Now, as I mentioned, the X-Clarity system has a couple of different modes. So the electrophoretic mode, or the ETC mode, and the passive mode. Now, we will concentrate on the electrophoretic mode, but you also just know that if you want to do passive clearing with the X Clarity system, that may be done. As a matter of fact, we have many customers that will potentially do an initial active clearing run and then set up for a couple of passive clearing runs is how they found the best to optimize their clearing samples. Now, as I mentioned earlier, there's no real difference in the protocols that you use based on your temperature or based on your tissue type. So the only real difference in tissue type is time. So, for example, a whole adult mouse brain may take anywhere from 7 to 10 hours, while the heart may take up to 24 hours. Where the differences come in is whether or not the sample is endogenously labeled or not. So if your sample is endogenously labeled, then we do have a set of current and temperature that we recommend that you use. However, if samples are not endogenously labeled, but will need to be antibody labeled, then you can raise these temperatures, increase the amp and raise the temperature, and this will speed up the process maybe by an hour or two. But the reason why we have these set so lower for the endogenous labels is so that they don't cook off the fluorescent signal. Once that's done, and the next is the tissue clearing. Now, this is a very important step also, in that what it does is that you have these samples that have been sitting in SDS for a while, and so you want to make sure that SDS is completely washed out, because if it still remains, then it has the potential to inhibit your downstream reactions, antibody labeling if that needs to be done, and potentially also to interfere with downstream imaging. After that, labeling if you need to do it, the refractive index matching, and then imaging, and then finally analysis. All told, with the system, the entire process from sample prep to imaging can take as little as a week for endogenous labeled samples, or probably up to about 3 weeks if you have large samples that need to be antibody labeled. The various sample holder options we provide allow you to process up to 6 or more larger tissue samples in one batch, depending. So that's the X Clarity system and a brief introduction to the process that goes around it. All told, so if we go back and we're still trying to figure out, okay, is this something that we want to do or not? Let me just put it this way, is that we view the tissue clearing universe a little bit differently than most others. To us, there's active clearing with X Clarity system and passive clearing that encompasses just about everything else. Admittedly, X Clarity is not suitable for all clearing applications. So we have some questions that we think will help you as you try to figure out what may be the best approach for you. And we've gone on over these, but I definitely think that they're worth repeating. And so that's, first, how compatible is your method with your imaging system and your objectives? And do you have the imaging system and objectives that you need to do this? Will tissue clearing remove or negatively impact your analyte? Will the tissue clearing sufficiently clear the volume that you need? Some tissue clearing methods do well with samples that are, say, from 500 microns to over a millimeter or to just about a millimeter. How will tissue clearing impact your labeling method? And what sort of throughput will you need? In other words, will tissue clearing be something that you do rarely on an occasional basis and you won't have very many samples? Will this be something that you're actually looking to set up as a sustained workflow for your lab moving forward? And so to answer the question of whether to X Clarity or not to X Clarity, in our view, we think the X Clarity system combines the speed and the power with the inherent flexibility built into the Clarity method. Where we have found the X Clarity to be the most successful is where the samples will be large in size and quantity to be cleared. The sample types may be diverse and labeling type may vary. Conversely, if the need for clearing will not be greater sporadic in frequency or tissue size is generally under a millimeter, we highly recommend that you initially investigate using some passive clearing method. Regardless, the only true way to determine what is the best method for you to use for your purposes is to learn by doing. Therefore, we strongly encourage you to test multiple methods to determine what will be best for your lab. So, to wrap up my portion is just to remember is that tissue clearing is just one component of that larger analytical pipeline. Your end goal is to acquire informative and insightful in-depth 3D images, and your other factors such as your goals and hypotheses, even your personnel, which we didn't touch upon, your lab labeling plans, and your sample type, and most especially your imaging system, all will bear on this tissue clearing strategy. And so with this, I am going to kick it back to Gulpreet to walk you through the considerations for imaging and the Olympus systems themselves.
Thanks for your wonderful talk, Tim. In his talk, Tim mentioned that for your experiment to be successful, sample clearing and imaging technologies must be compatible. We'll now delve into the imaging side of it. Now, as you already know, confocal and spinning disk microscopy both use epifluorescence geometry. In that, one objective is used to send excitation light to the sample and collect the emitted fluorescence. Optical sectioning is achieved by the presence of pinholes. However, the entire thickness of the specimen in the Z-plane is still illuminated with light multiple times, which can cause photobleaching. The sample is scanned one point at a time, rendering acquisition of volumetric images very long. Now, the speed of imaging in confocal microscopy can be improved by the presence of resonance scanners or gas photomultipliers, or by simply using a spinning disk configuration. However, faster scanning means fewer fluorophores are excited and fewer photons are emitted, so the signal-to-noise ratio is reduced. So confocal microscopy will provide superior Z resolution, but at the cost of speed and excitation efficiency. And it will work well for fixed tissue slices of limited thickness and time-lapse imaging of selected areas in living samples. But high-resolution imaging deep into biological tissues is not very efficient. In Lightsheet microscopy, the specimen is excited locally with a thin sheet of light instead of a focused spot. The emitted light is detected immediately by a detection objective, which is perpendicular to the illumination axis. Optical sectioning in this configuration is achieved at the illumination plane because only fluorophores in the illumination plane are excited, and there is no signal from fluorophores above or below this plane. This allows for high spatial resolution while performing volumetric imaging. A 3D stack of images can thus be acquired by illuminating each plane only once. This reduces the energy load on the fluorophores, resulting in reduced photobleaching and phototoxic effects. Now, this configuration of Lightsheet microscopy is called selective plane illumination microscopy. Next, I'd like to talk about multiphoton microscopy. In two-photon microscopy, which is the most common form of multiphoton microscopy, two photons are absorbed by the label at virtually the same instant. Multiphoton microscopy also uses longer wavelength photons, which are lower energy and therefore penetrate more deeply, creating less tissue damage while imaging further into a sample. However, just like in confocal microscopy, the sample is scanned one point at a time, making acquisition time really long. This makes it unsuitable for large samples or live tissues, which need to be captured very quickly. Now, here is a chart that compares various parameters among the technologies we just discussed. Let's start by focusing on the first row. As you can see, multiphoton systems are more complex and therefore pricier than confocal, spinning disk, or even Lightsheet microscopes. But while confocal and spinning disk systems have gotten to a point where you can have automated workflows and are super easy to use, multiphoton and light sheet systems still require some more understanding of sample and microscope behaviors. Where light sheet really shines is in the speed of acquisition. The acquisition speed is higher than even spinning the systems and much, much higher than most confocal and multiphoton systems. Consequently, sample degradation during imaging is much lower in light sheet systems and in spinning disk systems. Multiphoton systems do not photobleach the samples as much, but they can burn the tissues if exposed for too long. So you do have to be careful while using this workflow. On the other hand, confocal systems use a point scanning approach, so they tend to photobleach the sample quite a bit. Yet they do have the best sectioning effect, given that they have a proper pinhole that cuts out unfocused light. Now, spinning disk systems also have a set of pinholes, so one could argue that they have a similar sectioning effect as confocal systems, but they do suffer from a little bit of crosstalk between various pinholes, which is why the pinhole effect is not as remarkable compared to confocal systems. However, when you look at penetration depth, confocal and spinning disk systems do not perform as well as multiphoton and light sheet systems. So, overall, while confocal and spinning disk systems are well suited to image smaller samples at very high resolution, multiphoton and light sheet systems image larger samples better. I would like to point out that this chart is based on a generalization of technology, and what imaging technology you need to use will be dictated by not only your sample type, but also the experiment that you want to do. So let's look at some of these sample and experiment types in detail. Okay, when it comes to large cleared samples, such as whole organs, organoids, and tissue sections that are greater than 500 micron thickness in Z, light sheet microscopy will provide several advantages, such as deep penetration, fast acquisition rate, and low phototoxicity. However, light sheet microscopy in its SIM configuration is optimal for cellular to macro resolution. If you do want high subcellular resolution, that will be provided by confocal microscopy, but it will have high phototoxicity, slower acquisition, and low penetration depth. Multiphoton, on the other hand, will provide deep penetration and subcellular resolution to a certain extent, although it will take much longer. So really, if you want to image large samples, your best bets are light sheet or multiphoton systems, although light sheet will give you much faster outputs compared to multiphoton systems. This image shows a cleared whole mouse brain on alpha-3 light sheet microscope at 10x magnification. The top image is a volume rendering, whereas the bottom images show a Y and a Z projection. In this case, the researcher was interested in studying processes at a macro level. Now, when considering large sample images, you must consider your experiment type too. Do you really need to image the whole brain? If you just need to study one section of a fixed brain, you can simply get away by cutting it out. This will make your sample processing and imaging much, much simpler. This leads us into smaller samples, such as cells and spheroids. This is where confocal microscopy mostly shines, as it can handle slides and culture dishes very, very easily and provide subcellular resolution. However, if you do want to image one point for many, many days, for example, if you're doing cell migration assays, you might still consider light sheet microscopy, as it will provide you with fast acquisition rates with minimal phototoxicity. Yet this will limit your resolution limit. I have seen many researchers do calcium imaging studies on light sheet systems or even on multiphoton systems if the samples are slightly larger than what confocals can accommodate. So that leads us into the medium-ish size samples. I think of these as samples that lie somewhere between whole organs and cells. Consider whole zebrafish or plant roots. Now, the images in the right side panel show a cleared zebrafish brain at 10x magnification with alpha-3 light sheet microscope. You can observe neuronal staining and rainbow false coloring, and the bottom two images present zoomed-in versions of these images. Now, in these cases, light sheet microscopy, again, has the advantage of allowing one to image these macro processes in whole organisms with deep penetration and at a fast rate with minimal photodamage. But in some of these cases, confocal microscopy might just suffice. So if you're only interested in imaging the tail of a zebrafish at high resolution, you might consider confocal microscopy. And this loops us back into how your experiment is as important as the sample type. Now, going back to our focus today on deep tissue imaging, I would like to emphasize the benefits of Lightsheet microscopy in a SPIM setup for deep tissue imaging. It provides you high 3D spatial resolution, high signal-to-noise ratio, fast imaging acquisition rate, and low phototoxicity. Finally, let's discuss a few imaging considerations while imaging cleared samples. These include refractive index matching of detection optics with the sample media and sample mounting preferences. I would like to bring your attention to the fact that there are several different clearing methods, each with a different refractive index as noted in this table. And it is essential that detection optics are compatible with each of these clearing methods. Now, in the next diagram, you will see that when refractive index of an objective is higher than the refractive index of the sample, the PSF of the resulting image in XZ will appear to be elongated. On the other hand, when refractive index of the objective is less than the refractive index of the sample, then the resulting image will appear abnormally flatter. Perfect reconstruction is only achieved when refractive index of the objective perfectly matches the refractive index of the sample. Therefore, it is essential to use detection of optics that perfectly match the refractive index of the sample and the media that it was cleared in. Now, using gradient index lenses that allow you to tune the refractive index according to the media would be the way to go here. Shown here is a list of Olympus optics that are used for multiphoton and Lightsheet imaging. As you can see, they span the entire range of refractive indices from 1.33 to 1.52. They have correction collars that can be used to adjust the refractive index. And another thing you should note is that detection optics for Lightsheet microscopy has to have a long working distance, so you can actually image through the entire sample. All right, so the key points here are the detection optic that you use must have a long working distance and should have gradient index to allow you to match the refractive index of the sample and the clearing method used. Finally, I'd like to talk about some sample mounting requirements. Now, sample mounts for imaging large samples include various systems such as hydrogel embedding, hooks, or polymers that have good optical qualities. Basically, 3D samples require 3D sample mounts. The sample has to be held such that its 3D volume can be easily imaged. So the sample must be accessible from the top and at least one side for a Lightsheet system. And some sample holders will allow for manual or automated rotation of the sample to allow imaging from multiple angles, but it is absolutely essential that the sample holder be mechanically stable. If you're using fixed, cleared samples, the sample holder should also be able to hold the sample in refractive index matched media, such as the X-Clarity system. Finally, the sample mounting system needs to be easy to use because you're going to do this over and over. Now, on the right are some examples of mouse brain and kidney that are held in alpha-3 sample mounts. Now, finally, to summarize today's talk, I would like to mention a few points. The first is that tissue clearing is critical to increasing transmittance and penetration depth of large samples. There are several protocols available to do sample clearing, including X-Clarity from Logos BIOSYSTEMS, which Tim introduced us to. When imaging these samples, it is essential that you match the detection optics to the refractive index of the cleared sample. Finally, we spoke about many imaging technologies today and the advantages and disadvantages of these in several scenarios. For voluminous and cleared sample scenarios, we noticed that Lightsheet technology provides significant advantages. I'd like to just play the video again. This is a whole mouse brain that was captured with the alpha-3 Lightsheet microscope at 10x magnification. Well, with this, Tim and I would like to thank you all for your attendance and interest. We are happy to take questions. And at this point, I would like to introduce Dr. Joanna Hoverlook, who is a product manager for the Alpha 3 Lightsheet microscope, and she can help moderate some of these questions.
Thank you, Tim and Gulpreet, for a lovely presentation. While we have questions coming in, I have some already here for us. The first one's for Tim. How does the X-Clarity system affect the sample quality and structure compared to other clearing methods?
Very well. So with the X-Clarity system, so all clearing methods will impact your sample size and structure. With the clarity-based methods or most aqueous-based methods, what will happen is that you will get some increase in sample volume size. This will certainly be noticeable after you clear. And if you think back to some of the 3-panel images we always show before clearing, after clearing, and then after final index matching. Now, with the final index matching, the samples will come back down in size, maybe not to what they were before you actually started clearing, but they will reduce back down into size.
Thanks, Tim. That's really important for our customers to think about, especially when they're looking into their sample holders. Gal, the next one is for you. When using corrosive or toxic clearing medias, what considerations should be taken with these samples when imaging?
Yes, that's a great question. There are many different clearing methods out there, and some of them have very corrosive media. The most important thing would be to make sure that the sample holder that you're using can actually support that. For example, in our system, the chamber that holds the sample media is made of entirely steel and glass. And the glass is held on the chamber, on the steel chamber, with a silicone-based glue, so it doesn't melt even if you put in a very corrosive media. That being said, my understanding is like the X-Clarity system, the media is not very corrosive, so it doesn't really need that much attention in that sense. So yes, you should absolutely look at what type of media you're using, and then there's other stuff like just in general usage. Make sure you're always wearing gloves. Make sure it doesn't get in your hands, doesn't spill over, because it could be really toxic to even the microscope components. And then another consideration is that if you're dipping your objectives into such corrosive media, you might want to consider furnishing caps, which is what we try to do if we have to do that. So those are some of the considerations.
Thank you. And not seeing any other questions. If you have any questions, please feel free to type them in the chat. We'll be available for a few more minutes to answer your questions. But in the meantime, if you have any further questions, please feel free to reach out to Gulpreet or Tim at info@logosbio.com. Oh, we have a new question. This one would be for Tim. What would be the optimal concentration of antibody to label whole mouse brain that's cleared?
Again, I would not recommend trying to label a whole mouse brain. I would consider working in hemispheres or even smaller slices. And so one of the things that you could do is with the sort of clarity-based method is you can slice your sample anytime prior to clearing. So if you are going to work in smaller samples or sections, you'll want to slice it before you actually start the removal, because once the lipids are removed and you slice after that, you tend to get induced artifacts and tear up your tissues. Now, as far as antibody concentrations, it's a pretty high concentration. So, for example, if you have a general 1 mg per mil stock of your antibody, so for your primary, you're going to want to use anywhere from about a 1 to a 50 dilution to maybe a 1 to 250 dilution at most. But more concentrated is better. But you will also need to work that out. That is one of the things that you will need to optimize is versus concentration versus labeling time versus background signal and things like that. And so that's why we also recommend starting off with smaller slices, especially if you're going to be doing antibody labeling, so that you can work all that out without a huge amount of time or expense with the reagents.
Thank you, Tim. And yeah, it's great to know that we have you as a resource and our customers have you as a resource, because sometimes just finding a protocol in a journal article, you don't have somebody to go to really help with that.
So there's one other question in the chat panel. Not sure you guys want to take that question now. Talking about sample size, trying a rat brain hemisphere to identify a connection spanning 3/4 of the brain. Clarity cubic, etc. spans so much, so I had to go with F-disco, V-disco, etc. Solvent-based protocol is the one I'm trying now. Huge background with nanoboosters and permeation are a major problem. Any recommendations?
Yeah, so with the rat brain, we have seen that. And so I guess if you do info and just ask me and put that question in there, so I would expect the rat brain, yes, it is going to swell. And as you saw with the solvent-based methods, samples tend to reduce. And some of those consequences. And if you email us, what we can do is we can get you a more optimized protocol for that.
Sounds good. Well, if there are no other questions, well, I guess we will wrap this up. I want to, on behalf of Olympus, I'd like to thank all the attendees for joining us, and Drs. Carr and Stepp for their expertise and participation in today's event. We hope this material presented was informative and useful. This webinar will be archived on our website at www.olympus-lifescience.com. Everyone who registered for the event will receive a follow-up email with links to the archived presentation, whether you attended today or not. That's going to do it for today. Thanks again for your interest in taking the time to attend this Olympus event, and we will see you again next time.
Thank you.
Thank you, everyone.